Welcome everyone to the C-DEBI Network Speaker Series. This series started in November 2011, and the first person who gave a talk was none other than Brandy Reese. And she happens to be the PhD advisor of our speaker today, who happens to be the last of the speaker series. Look for an email from C-DEBI in the next couple of weeks. We are going to have a different communication type of format that will come from Rosalind in the next week or two to describe how we're going to be doing that. But this represents our last speaker in the series. And today I've got the honor of introducing Megan. And she got her bachelor's degree at the University of Southern Mississippi, and then went on to, she's right now finishing up her PhD at Texas A&M Corpus Christi. I got to meet Megan out at sea. We did a trip to the mid-Atlantic, a nice 30-day cruise out there to collect some samples. Today, she's going to be talking about some of the results from that expedition. Also, I was part of an expedition to the Mariana forearc and got Megan some samples, and she will also be talking about some of those. And those are pretty cool in the sense that those samples are from a serpentine seamount. It's the only place on Earth where I know where there's naturally occurring blue mud. And so Megan will be putting together both of those for us today in her seminar. So I look forward to it.
All right. Thank you, Jeff. I appreciate the introduction. As he said, my name is Megan Mullis, and I'm really thankful to be here today to share two of my dissertation chapters with you all. And so the title of my talk is Outwit, Outplay, Outlast, a microbial tale of survival in the marine deep subsurface environment. And I'm sure some of you noticed the logo at the top. This is a tale of survival. So it's basically the microbial version of Survivor. Before I dive into the talk, I want to give a brief outline of where I'll be going in this discussion today. It'll begin with an introduction about how we actually investigate and analyze microbes within deep sea sediments. And we use a two-pronged approach. Specifically for my dissertation research, we use this approach. And so the culture-independent methods was used for the Mariana forearc sediments, and the culture-dependent methods were used for the western flank of the Mid-Atlantic Ridge sediments. And I'll get into both of those projects and the results and future directions from these studies. So let's start from the very beginning. What are microbes and what are they doing? Two essential questions that guide the marine subsurface exploration. Microbes are found in all three domains of life, including bacteria, archaea, and single-celled eukarya. They're found in nearly every conceivable environment, and they have various mechanisms for survival. And many of us have probably seen this schematic from Outwit et al. In 2011. And this is just illustrating the vast amount of environments that these microbes can be found in the marine subsurface. Microbes are in fact so numerous within our marine subsurface sediments that they outnumber the stars in our sky. So with that overwhelming amount of biomass, there is many questions about what these microbes are doing and the diversity of these cells. In a basic essence, microbes need four things to survive. They need water, some type of nutrient, electron donors, and electron acceptors. And if these microbes have access to all four of these components, they can withstand that natural environment. But they can also have various survival mechanisms within their natural environments. The first being that these microbes can adapt to their surrounding environment. So they can utilize the resources available to them and be better fit for that natural ecosystem. The second is they could form mutualistic relationships with one another. So one population of microorganisms could utilize carbon dioxide and create methane, while another population could utilize that methane and then create carbon dioxide, creating this loop effect. So it can be a symbiotic relationship. The third mechanism is a competition mechanism. And the two common methods for competition is inhibiting the growth of the surrounding neighbors to gain access to those resources for survival, or they can in fact kill or lyse those surrounding cells to gain access to those resources or even utilize that lysed cell for nutrients and resources. And many of us are familiar with this competition mechanism because at some point in our life, all of us have taken an antibiotic for some type of infection. Antibiotics are actually secondary metabolites that are naturally created from microorganisms. And I have here a schematic from a paper that we published 2 years ago. And to basically describe this schematic, we have 2 antibiotic genes that are very common for producing these toxic-like compounds. And these genes are the polyketide synthase gene and the non-ribosomal peptide synthase gene. They're abbreviated here PKS and NRPS. And there's a hybrid gene below composed of these 2 genes. And essentially this image here is illustrating this gene structure for antibiotics and the resulting compound from this gene that these microbes can produce. In this study, we previously looked at existing data sets from various marine environments and terrestrial environments from the deep subsurface. And the marine subsurface environments we used were the western flank of the Mid-Atlantic Ridge and Juan de Fuca Ridge for this investigation. And we looked in these data sets for the PKS and NRPS genes to see if these antibiotics were present. And these 2 phylogenetic trees are essentially showing with all of these blue dots that there are antibiotic components within these marine and terrestrial deep subsurface environments. And the main takeaway from this slide here is that these marine environments contain potentially novel or unique antibiotic genes that haven't been looked at before. They're distantly similar to those terrestrial analogs, but they could be unique and a unique necessary or a unique survival mechanism. Now, how do we go about analyzing these microbes within these environments? It's known that the majority of microorganisms within a natural environment are very difficult to culture in the lab, and we have no cultured representatives of them. And that can be due to the lack of resources we can provide for them in a laboratory setting. They could actually be needing a co-culture where they need to grow with a fellow microbial population, and we don't know that. We could also just not replicate the exact culturing conditions they need to survive. So that leads to this 2-pronged approach to analyzing microbial communities. And I'll briefly explain this 2-pronged approach. The first part is this culture-independent method. So culture-independent methods focus on taking an environmental sample and isolating that genomic content, in this case being DNA, and sequencing those DNA molecules, assembling them, and annotating them for comparative genomics. And then culture-dependent focuses on actually culturing those individual microbial cells and examining its ecophysiology in a laboratory setting, and possibly running comparative genomics from that approach. So this 2-pronged approach development idea was in Brandy Kilreese's lab, and we utilized this novel high-throughput culturing technique to sort individual cells into a sterile media source and have them grow to analyze their physiology as well as their genomic content. And I'll get into that later in the talk. This is a simple, or not a simple, but a overall phylogenetic tree of the bacteria, Archaea, and Eukarya. And this is from 2016. So it's definitely got some more taxa that should be populated here. But the main takeaway from this is that all of these red dots on the edges of these branches here illustrate taxa that have no cultured representatives. So all that we know about these taxa is from a culture independent standpoint. And so this culture dependent method that I'll introduce later in the talk could be useful in isolating these individual taxa that we know very little other than their genomic content. So this could actually really help populate this tree of life. And the last part of my introduction is focusing on some terminology, so that way all of us that are attending today can be on the same page. So when I talk about genomic content, I'm referring to DNA and RNA molecules. DNA is a double-stranded structure that contains all of the genetic blueprint for a single organism. So essentially, it encodes all of the genes that microbe could use throughout its life. RNA is derived from DNA, and we use it as a proxy for activity. RNA is transcribed from DNA to then go on to be translated to make proteins. And we use this RNA in order to determine who is actively trying to function in that community. A genome is the collection of genes for a single organism. A transcriptome is a collection of the total RNA for a single organism. Metagenome is the collection of genes within a total environmental sample. So that can include multiple microbial populations. Metatranscriptomes are the total RNA collection from an environmental sample. So it could be the total RNA from multiple microbial populations. And I italicized genome and metatranscriptome here because these are the two methods we used in these studies. Now, the first portion of my talk is going to focus on the Outwit component. So looking at the microbial function as a method for survival. And this component focuses on the Mariana convergent margin. The Mariana convergent margin was sampled on the IODP expedition 366. And you can see the actual transit and sample map here on the left. On the right, this is a schematic illustrating the Mariana region. And the region that was sampled was the 4-arc region, which you can see illustrated by this yellow star here. And so on the left in this schematic, I have a bathymetric map illustrating the chain of seamounts that were sampled on this expedition, labeled with their names and the IODP sample numbers here. The seamounts that were sampled were Asut-Dasoru, Fantagisnia, and Yenazao seamounts. And on the left, this is a horizontal depiction of the actual Mariana 4-arc. And I just want to orient everybody briefly with this system. Most of us are familiar with the Mariana Trench being the deepest point on Earth, and that occurs from this subduction zone process. So essentially, the Pacific plate is being subducted under the Philippine plate, and in that subduction process, sediment and seawater is being subducted under that Philippine plate as well. Upon that seawater interaction with the mantle layer, serpentinization occurs. And this serpentinization allows for these serpentinite molecules and compounds to then travel through that conduit illustrated through these red arrows here to the tops of these seamounts that have formed. And you can see these seamounts illustrated here, again, Asut-Dasoru, Fantagisnia, and Yenazao seamounts. And so these are serpentinizing environments, which are very important because they release a high amount of hydrogen gas, methane gas, and can really increase the pH in this system, which in one of the seamounts, Asut-Dasoru, the pH reaches 12.5, and 12.5 is the actual limit of life on Earth for pH that we know of. So if we found microbial communities actively trying to function within this site, it can be really telling about how they can withstand this extreme environment. And so we predicted that this Mariana 4-arc system was going to be important for hydrogen metabolisms as well as methanogenesis and methanotrophy. And this image here is from the actual IODP expedition, and you can see these beautiful blue-green colors of the sediment cores are indicative of that active serpentinizing reaction that Jeff mentioned at the beginning of the talk. And I wanted to provide some of the geochemistry that was collected on board the expedition. And to orient everybody with this graph here, we have methane and sulfate concentrations as well as pH on the x-axis. And we have depth increasing in meters below seafloor on the y-axis. And you can see all of the sites here listed in the legend. And we have some key notes that I want everyone to notice. Asut-Dasoru is in this red square, and you can see that the methane concentrations are really high in certain depths, up to 5 millimolar in some cases. And again, that pH is corresponding to that high methane release from that serpentinizing reaction in the Asut-Dasoru summit. So this is further indicating that that serpentinizing reaction is actively occurring in a much higher fashion in Asut-Dasoru. And I want to briefly describe the methods we used for this study. First, the sediments were drilled and collected from December 2016 to February 2017 aboard the JOIDES Resolution. And those sediments were stored at -80 degrees Celsius until the extraction process back at the Reese Geomicrobiology Lab. The total RNA was extracted using a hot alkaline lysis protocol adapted from Morono et al. In 2014. and the total DNA was removed from sequencing because we wanted to look at the total RNA, so the total activity from those microbes within those sites. So we sequenced the total RNA using Illumina NovaSeq and processed those reads and annotated them against many databases for gene expression analysis and predicted potential metabolites that these microbes could be forming. Before I get into the actual results from this study, I want to take a minute and talk about contamination controls, because we take contamination in the lab very seriously to not contaminate our samples. So first, we included the drilling fluid used on the expedition and no template controls alongside all of the Mariana 4-arc samples in the lab. So we extracted them alongside our samples, processed them, and sequenced them alongside the samples. From those sequences, the reads that were assigned to the contaminant samples were then removed from the Mariana samples using BB split. So essentially, we aligned the Mariana reads to those contaminant reads, and if they matched, they were removed from downstream analysis. And finally, the taxa that remained from that contamination removal that are commonly associated with contamination was then analyzed using a Bray-Curtis distance for a non-metric dimensional scaling ordination, which you can see here. And we expected from this ordination that if the contamination was across the board and it actually was contaminating all of these samples, that they would be tightly clustered together. But you can see here, there's no clear clustering of these samples, and there's actual geochemical drivers trying to explain that diversity we found. And this idea was an excellent idea from Karen Lloyd and Brandy Keel-Reese in our many discussions about preparing our samples. So after contamination removal, these are the taxa that we found within the Mariana 4-arc. So on the top left, we have Archaea, which was in much low relative abundance compared to Eukarya and bacteria. The Eukarya we found was dominated by Opisthocanta, which is mainly comprised of fungi. And we would expect that this is the finding because the fungi could be actively sporulating within that Mariana 4-arc system to withstand those extreme conditions. The bacteria we found to be dominated by Pseudomonadales and Burkholdeриales. These two orders have a wide range of metabolic capabilities, so it would make sense that they are in such high relative abundance within these sites. And we also found that unknown bacteria comprised anywhere from 10 to 15% of our samples. So the next thing we did for our analysis was to examine those geochemical drivers that could be explaining the taxa diversity as well as the functional diversity. And so how we did this was we ran an ANOVA on the geochemical drivers and created a canonical correspondence analysis. And we found that the geochemical drivers that drove taxa diversity was lithium, bromide, ammonium, phosphate, and sulfate, while the drivers for functional diversity were lithium, bromide, silicon, iron, and magnesium. The main takeaway that we found from these two analyses were very similar results. We found that all the summit sites from all three seamounts, regardless of what seamount it was, clustered tightly together. So they were the most similar to one another. While the flank sites, the sides of those seamounts, were significantly different from each other and significantly different from the summit sites. So that was a key finding across the board. The majority of the reads annotated for the Mariana 4-arc were in three categories overall: for metabolic process, catalytic activity, and unclassified and hypothetical proteins. And you can see here that this relative abundance chart, the darker the color means the more gene expression for that, that category. And so you can see that there's a massive amount of expression for metabolic process and catalytic activity. So to further dive into that metabolic category of metabolic activity, I hand curated redox tower genes located in that gene column here, and annotated the metatranscriptomes against those genes to see how much level of expression there was, and normalize that read count to transcripts per million. And you can see here the basic breakdown is carbon cycling, hydrogen cycling, oxygen, denitrification, and sulfur reduction and oxidation. The sulfur reduction and oxidation was very low relatively across the board for dissimilatory processes. Assimilatory sulfur use was more common among all the sites. So we think that they were more concerned with creating proteins rather than breaking down these compounds of sulfur. And the main trend we found from these data was that methane oxidation and denitrification were highly expressed across the board, regardless of summit or flank sites of these seamounts. And to put that into perspective here, these bubbles are corresponding to the transcript per million. And you can see in this bottom left image that the anaerobic methane oxidation and partial denitrification were highly expressed, again, across the board. And so we think that there's this coupling reaction of these two metabolisms because anaerobic methane oxidation is not as energetically favorable as other metabolisms. So a microbe that's performing this metabolic reaction is not going to get the energy payoff that it would want. So we would think that this denitrification coupling with that anaerobic methane oxidation could be maximizing that energetic payoff with those microbes. And again, the sulfur cycling here is illustrated, and we found that dissimilatory was very low in gene expression when compared to other cycles. So overall, we found that the microorganisms within the Mariana fork are active and thriving, and we hypothesized that there's this coupling reaction of denitrification and anaerobic methane oxidation. We also found that the flank sites were significantly different from the summit sites in both microbial diversity and functionality. And we think that could be due to the active serpentinizing reaction occurring within that conduit to the summit sites, while on the flank sites, they're not receiving that much of an active serpentinizing reaction. It's also probably more oxygenated than those summit sites. The next portion of my talk is going to focus on outlasting your neighbor. So we recovered isolates from the western flank of the Mid-Atlantic Ridge and characterized their genomes. And this is the culture-dependent approach. So just to familiarize ourselves a little bit with the western flank of the Mid-Atlantic Ridge, it's also commonly referred to as North Pond, so I'll refer to it as North Pond from here on out. This site is a shallow sediment pond overlying a basaltic crust with active fluid flow. And on a previous IODP expedition, the sediment was drilled down to the basement, and these cork observatories were installed to monitor the microbial diversity and activity over time. And so in 2017, there was a return expedition to North Pond that I sailed on to collect these basaltic fluids and these sediments. And our primary goal was to try and isolate individual bacteria, archaea, and or fungi. And we tried to do this through a novel approach. The approach we used was a single cell sorting method for high throughput culturing. And how we did this was this method was developed and conceived from Dr. Brandy Kilreese and Rachel Wiesand. And so there were many tests to run if we could sort individual cells into a sterile medium and grow them over time to perform ecophysiology tests or genomic characterization. And so we utilized fluorescence activated cell sorting, FACS, using this instrument called the FACS-JAZ. And we utilized a cell viability kit that contained 2 fluorescent dyes to stain the microbial cells. And these dyes are the propydium iodide dye and thiazole orange. Propydium iodide is a cell membrane impermeant dye, so it needs to enter the contents of a cell through some kind of hole or if the membrane is lysed. A thiazole orange dye is a cell membrane permeant dye, so it can pass freely through that membrane. And that's how we can distinguish if that cell is viable or not. We were looking for if that membrane was intact. So we would stain the cells with these 2 dyes, and based on that membrane intactness and fluorescence that it gave off for the machine, 2 lasers would sort that fluorescence level, and we could sort those cells that are viable into a selective media. So the samples we collected were collected aboard the Atlantis in October of 2017, and we accessed the boreholes and shallow sediments through the remotely operated vehicle, the Jason. And we collected shallow sediment cores, 0 to 10 centimeters in this case, and the basaltic fluids, and we took those fluids and sediments and enriched them in various media types and stored them at 4 degrees until we cell sorted them. In the sediments, we added a gram of sediment from each 2-centimeter increment of the push core into a sterile medium and created a sediment slurry. From those sediment slurries, we separated the cells from that sediment matrix by gently centrifuging them at a low speed to prevent any cell breakage. Within that supernatant that's separated, we stained those cells using the propydium iodide and thiazole orange dyes and prepared them for cell sorting. We also included sterile media in this process to not only track contamination, but calibrate the instrument in case there was some noise from any particulates within that media, as well as if there was contamination. And so we prepared it the same way for cell sorting and prepared for gate selection. Now, gate selection is how we actually calibrate the instrument for cell sorting. And so based on those fluorescence levels from those samples, we can plot the forward scatter and side scatter of fluorescence. And in this image here, each dot represents each event or each cell, theoretically, that passes through the lasers. So in the bottom left, you can see this phosphate buffered dye, and the purple dots correspond to just that phosphate buffered saline that's been stained. So that's the noise that we don't want to select for. The non-viable region is the region that has that propydium iodide dye that's telling us that those membranes aren't intact in some way. So we don't want that region either. The viable cells region is the region that only has that thiazole orange dye, and that's what we want sorted into our media to grow. And so my lab mate, Rachel Wisen, ran many iterations of this test to validate that it could be possible, and she worked extremely hard on this process and found that these sorts were successful to some degree, and we sorted 1, 5, and 10 cells into various media and calculated its success rate, and also statistically tested using t-tests to see if it was significant. And so from those results, we calibrated it for the North Pond sorts, and I have a schematic here in panel B illustrating the cells that were sorted into the sterile media. And so these colors are corresponding to the number of cells that were sorted into each well. And it was 1, 5, and 10 cells that were sorted. In those gray wells are no cells were sorted. So we use that as our no template control to track contamination. And we measured optical density daily at 595 nanometers and plotted those absorbance levels to generate a growth curve. Based on those growth curves, we found that the 1 cell success rate was 81%, 5 cell success rate was 75, and 10 cell was 52. And these are the actual growth curves from this test. And we have on the left here, 0 to 2 centimeters, and on the right, 4 to 6 centimeters with time on the x-axis and absorbance on the y-axis. And you can see that over time, it generates this beautiful growth curve for 1, 5, and 10 cells. And so we definitely observed some microbial growth. The no template controls were a big sigh of relief because the wells that did not have anything sorted did not approach 0.2 for absorbance, which is usually the threshold for microbial growth. We also included a no template control plate of sterile media. And again, all 96 wells illustrated no growth. So we potentially have no contamination. Based on the isolates that had the most growth, we calculated 46 to have the highest growth and do the best in this culturing condition. And we selected them for identification of Sanger sequencing for the 16S rRNA gene, and 30 of those isolates were able to be identified. And they were closely matching to Bacillus, Idiomarina, and Marinobacter. 4 of those sequences matched to an uncultured and an unidentified bacterium in GenBank. So we decided to make a 16S phylogenetic tree, and we selected 5 isolates for whole genome analysis. So we selected the one that closely matched to Bacillus, closely matched to Marinobacter, and then 3 that closely matched to Idiomarina. And based on those isolates, we extracted their genome content, sequenced it using Illumina MiSeq, and those reads were merged and the adapters were trimmed. Then they were assembled using velvet. The genomes were then annotated using various annotations. The genomes were checked for completeness using CheckM, which checks for the single copy marker genes. And we ran some comparative genomics afterwards. And so our overall finding of these whole genome isolates was that we had some unique strains, which was super exciting. So for the Idiomarina abyssalis strains, we found them to be, based on their phylogenies and their average nucleotide identities, to be 99.5% similar to one another, which indicates the same strain. So we named it Idiomarina abyssalis strain KJE after Katrina Edwards. On the right, you can see it's a phylogenetic tree for Marinobacter. And the two isolates, the bacillus isolate actually was misidentified, and it turns out it was a Marinobacter. But they were two unique strains, so we named them Marinobacter celarius NP 2017 for North Pond 2017 and AT 3901 for the expedition number. And I have on these trees some other labels in blue, and these are corresponding to metagenome assembled genomes from a previous North Pond study. So these isolates have been found in the basaltic fluids as well as the sediments and the water column above North Pond. So it would make sense that these isolates are true to North Pond. Based on the whole genome assemblies, we annotated those genomes using KEGG decoder to estimate metabolic pathway completeness. And so this is the overall breakdown of those pathways. I labeled a few of them here. On the right-hand side, you can see Idiomarina abyssalis, and then the Marinobacter strains on the left. And they all had, to some degree, oxygen respiration pathways, while Idiomarina had dissimilatory nitrate reduction. They all had, to some degree, a fermentation pathway, and Idiomarina abyssalis had sulfide oxidation, while the Marinobacter strain AT 3901 had sulfur assimilation and sulfur dioxygenase. And just to put that in a simpler schematic for everyone, this is an illustration illustrating the capabilities of those microbes. And so we have those Idiomarina genomes on the left. And again, we have the oxygen respiration, the dissimilatory nitrate reduction, some transporters across the membrane, and it's capable of creating flagellum and movement. The Marinobacter genomes, again, has oxygen respiration and nitrate reduction. They have more transporters across its membrane, the capability of creating flagellum and movement, and the capability of using the Ittner-Duderoff pathway. And the final portion of my talk is going to focus on the outplay dynamic using potential antibiotic production within these 5 isolates. So we took those whole genome assemblies from each of these isolates and annotated them against ANISMASH, which ANISMASH is a database that includes all antibiotic gene clusters that we know of. And it data mines the data set that you put for any antibiotic biosynthesis gene clusters. And so the output is this schematic here, which this is a schematic that's kind of busy, but it's essentially a gene organization for antibiotic biosynthesis. And so for all 3 genomes for Idiomarina abyssalis, we found that they have the capability to possibly produce 3 different antibiotic compounds. While the Marinobacter celarius strains, we did the same process. They were capable of having 4 unique antibiotic gene clusters, including that non-ribosomal peptide synthase gene that I mentioned at the beginning of my talk. So these could be used to create these metabolites that could be used for a competitive advantage. And strain AT 3901 was unique that it had a siderophore present, which siderophores are known to be used for iron transport across the cell membrane. However, the gene is very similar in genetic structure to an antibiotic gene. So this siderophore gene could be used for that iron transport, or it could also be used for some type of antibiotic biosynthesis mechanism. And so the overall results from North Pond was that we found this cultivation dependent method to be very useful and effective for comparative genomic work. Using this high throughput method of fluorescence activated cell sorting can select for specific microbial guilds or isolate those microbes in low relative abundance from omic data. As I mentioned before, Marinobacter and Idiomarina were present in the past North Pond data, but they were in much lower relative abundance within those data. And we were able to successfully isolate them using this high throughput method. And now that we have validated that this can work and we've isolated specific bacteria that are unique, you could use it to target specific microbial guilds like nitrate reducers or sulfate reducers, whichever group you want to focus on. And the single cell culturing method is really unique because it bypasses that batch or enrichment culturing technique of taking your environmental sample and putting it into a media source. Because when you put an environmental sample into that medium and have multiple microbial populations, they could be trying to compete with one another, and a bacterial weed that's better fit for that environment, that's easy to grow, could overtake that entire culture. So this process allows you to sort individual cells to prevent that from happening. So finally, from these two studies, we found that microbial diversity and functionality can be assessed through both a culture independent and dependent method. And I would even go as far to advocate using both of these methods for future exploration of the marine subsurface, because we can use this culture independent approach to survey the environmental system and examine that microbial diversity and functionality, and then use that approach for a targeted cultivation technique. So you could look for taxa that have yet to be cultured, or even those taxa of interest of some scientific questions that you may ask. And finally, microbial survival is of the most important within the marine subsurface. And in previous studies, we've surveyed microbial environments for microbial diversity and functionality, but there's been a very small amount of studies that have looked at survival mechanisms such as secondary metabolite biosynthesis. So I would advocate not only doing that survey approach of looking at the diversity and functionality, but also look for these special metabolite biosynthesis genes that could explain how these microbes could persist within these extreme environments. And before I end my talk today, I have many acknowledgments to make. I want to first and foremost thank my advisor, Dr. Brandy Keel-Reese, my committee members, the Reese Geomicrobiology Lab, especially Rachel Wiesen and Christian Cunningham, Dr. Julie Huber for having me sail on the North Pond expedition, IODP Expedition 366 Science Party and Crew to the Mariana Forark, AT 3901 Expedition Science Party and Crew to North Pond, all of the other collaborators for this study and people that I have pestered so many times to help me. I also want to thank all of the funding sources, including the Schlesinger Ocean Drilling Fellowship and C-DEBI, for not only the funds, but the support over my time as a graduate student. And thank everybody for attending today. I really do appreciate your time. And with that, I will be happy to take any questions that you may have.
Super. Thank you very much.
Thank you.
So if you have questions, yes, by all means, please enter them. I'm sure there's a few that will pop up. And while all the congratulations and nice talk comments are zipping through the chat box, I've got a question for you. So with the Mariana fluids, the fluids start at depths of 12 to 18 kilometers below the seafloor. Did you see any microbes that have that are common with pressure related microbes?
That's a great question. I did not see any of the piezophiles within the diversity data. And the deepest sample we had within the Mariana Forark was from 20 meters below seafloor in the Fantagisnia seamount in the summit. So every other sample within the Mariana Forark was relatively shallow, between 1 and 5 meters below seafloor, and then that one that was at 20. Okay.
I see there's lots of awesome comments coming through here. So while the awesome comments are, oh, here we go. First question from Julie.
No, sorry, it's super confusing how I just typed that. Hi, Megan.
Hi.
So
Yes,
yeah, I'll ask my question better. In the Mariana section, did you do amplicon sequencing, the phylogenetic data you showed, or was that inferred from the metatranscriptomes?
that was inferred from the metatranscriptomes. We did do a preliminary amplicon screen from 3 samples to make sure that we could get material out of these seamounts. And then we inferred the rest from the metatranscriptomes. And I agree with you that, we didn't detect many Archaea, and that anaerobic methane oxidation is definitely a key area of interest. So I'm definitely looking through that data to try and decipher better what's happening. Because originally when we were going through the annotations, the methane oxidation was not really appearing within those annotations because we used the metagenome analyzer against various databases. So I hand curated those genes to annotate the metatranscriptomes against. And I think that if we didn't look for that, we wouldn't have known it was there because there was such an overwhelming amount of methane that we wouldn't think that there was methane oxidation happening. So there's definitely more to the story. And so I'm definitely diving into that right now.
Awesome, thanks.
Mm-hmm.
I see Jason has a comment, question.
Did you have to do anything special to get enough RNA to sequence? We extracted and extracted and extracted. So we extracted all of the samples that had high cell count from the Mariana fork. And I say high loosely, there was very few samples of the Mariana fork that had a relatively high cell abundance. Most of them fell below the detection limit for cell counts. So we extracted the majority of those samples to the point where we had no more sediment to extract. And we sequenced them. And from sequencing, we kept them separate, but we didn't assemble the metatranscriptomes for in silico analysis because when we assembled, some of them failed and some of them lost a whole lot of data. So we really worked with what we had.
Thanks. So you're saying the analysis was done on the individual reads, not the. Okay, cool.
Yeah, we merged the forward and reverse reads, but from there we tried to assemble, and the assemblies were really not great, and in a few of the samples they failed. So.
Good. Well, it looked great anyway. Yeah, thank you. Nice job.
Thank you. Okay, we have a message from Dawn. I'm not sure if I saw this right, but it looked like most of the genes for methane oxidation were PMMO. If so, could you verify that the PMMO was affiliated with the NC10 clade? It's an excellent question. I should definitely look into that because the way I analyzed this was to look at those matches for PMMO. Specifically, I haven't tried to map them back to any clades. I just was looking at the overall abundance of them. Okay, we have another question from Kelly. I've got a question about your culture-dependent FACS works from North Pond. I noticed that you had a higher success rate when you sorted fewer cells. Any idea why that is? Just competition? It's a great question. We've definitely hypothesized that it could be a competition mechanism. It also is an imperfect sorting method because first, I should say that we are using lower concentrations of those fluorescent dyes that the company suggests because we don't want it to inhibit the microbial growth. And we also know that when we sort individual cells, like one cell, we've seen some be very unsuccessful. So we hypothesize that it could be mistaking a particulate or nothing or part of a cell instead of it actually being a viable cell. So it could have just been in this particular experiment that these 10 cells weren't as successful as the 1 or 5 cells. So that's definitely something we can look into with more replicates. And for this study in particular, we sorted 2 96 well plates for these isolates for North Pond, but we have an overwhelming amount of other data from previous sorts that we can dive into.
Do we have any other questions? All right, well, I think we'll thank you, and thank you for presenting the very last of these the C-DEBI Speakers Series. Congratulations. And I understand this is going to be most of your PhD dissertation talk.
Yes,
So,
it will
yeah,
be.
we look forward to you finishing. That's great.
Thank you
And
very much. If anyone has any suggestions, I'll gladly take them as well. So, but I appreciate everybody's time and attending today. Thank you very much.